Hello friends, I am Dr. Marathe from the Department of Microbiology, Shardabai Kaur Mahila College, Shardanagar, Bharamati. Today we are going to learn about the cycle known as citric acid cycle. This is also known as TCA cycle. TCA cycle. And it is also known as Krebs cycle. Why is this called as a citric acid cycle? The reason is citrate is one of the unique intermediate of this cycle. That's why it is called as citric acid cycle. Why is this called as a tricarboxylic acid cycle? The reason is there are few intermediates in this cycle which contains 3 carboxylic acid groups. 3 carboxylic groups in the structure or in the cycle or in the structure of those intermediates. That's why they are also called as, it is also called as tricarboxylic acid cycle. And third, why it is called as Krebs cycle? The reason then is what? This cycle, it was first elucidated by Hans Adolf Krebs in 1933. From 1933 or 1937, and for this he got a Nobel Prize in 1952 in the field of physiology and medicine. So, in honor of him, this cycle is also called as Krebs cycle. Okay, now what is the role of Krebs cycle? Krebs cycle actually it is involved for the catabolism of, or you can say oxidation of acetyl-CoA through it. Now this is the part of central energy pathway. Central energy yielding pathway. The question may rise in your mind, what is the central energy yielding pathway? Central energy yielding pathway, it starts with glucose. After 10 successive reactions, that glucose by the process of glycolysis, it is converted into pyruvic acid. Now that pyruvic acid or its fate depends on presence and absence of oxygen. If oxygen is not available, then pyruvic acid enters into the fermentation pathway and it is converted into lactic acid by the enzyme lactate dehydrogenase. But if the oxygen is available, then pyruvic acid enters into the TCA cycle. But that entry into the TCA cycle is through acetyl-CoA. Its structure is CH3, C double bond O, and bond SCOA. This is the structure of acetyl-CoA. Now this acetyl-CoA then enters into the TCA cycle. Okay, and its two carbon atoms of the acetyl-CoA, these are channeled through the TCA cycle, they are catabolized through the TCA cycle. Okay, and the energy is released. Okay, so glycolysis, this end product of glycolysis, pyruvic acid, it gets entry into the TCA cycle through one intermediate, that is acetyl-CoA, and from TCA cycle, electrons are removed. There are 4 steps in the TCA cycle where the electrons are removed. They are accepted either by NAD or FAD, gets reduced to NADH plus or FADH2. They donate the electron to electron transport chain. those electrons they traverse the electron transport chain and they releases the energy, and that energy is trapped in the form of ATP. And finally, electron goes and binds with the reacts with the final electron acceptor, that is called as oxygen, and there is the formation of water. So this whole thing, starting from the glucose to pyruvic acid to acetyl-CoA into the TCA cycle. From the TCA cycle, electrons are removed, they enter into the electron transport chain, finally they meet the oxygen, which is finally true. This is called as a central energy yielding pathway. So this TCA cycle, it performs a very key role in this central energy yielding pathway. Why this is called as a central energy yielding pathway? Because whatever the other reactions or whatever the peripheral pathways are there, so some of the intermediates of this peripheral pathway, they get linked to this central energy yielding pathway, and finally it is catabolized into carbon dioxide and water. So that's why I am saying TCA cycle, it is a key part of central energy yielding pathway. Now the question again will rise in your mind, where this cycle is located, or the enzymes which are responsible for TCA cycle or the catabolism of acetyl-CoA through TCA cycle, they are located in the mitochondria of eukaryotic cell. In prokaryotes, obviously it is present in the cytoplasm. Okay, so this is the most important thing. Now let us discuss about how this acetyl-CoA enters into the TCA cycle. So we try to learn it by step by step, and which enzymes they are responsible for each reactions. Okay, so we will have the structure of acetyl-CoA, that is CH3 C double bond O S COA. This is acetyl-CoA. Acetyl coenzyme A. Now this acetyl-CoA, this acetyl-CoA condenses with the oxidative, that is COOH double bond O CH2 COOH. This is known as oxaloacetate. Now this oxaloacetate, which is a 4-carbon containing compound, okay, which reacts with acetyl-CoA. The 2 carbon of the acetyl-CoA, they get connected to this 4-carbon structure, and it forms the 6-carbon structure, that is known as citrate. Its name is citrate. Okay. Now this citrate, it has a structure like this. COOH, COOH, CH2, COOH, CH2, COOH. This is called as citrate. Now this is the unique intermediate present in the TCA cycle. That's why this cycle is also called as citric acid cycle. Here, during the reaction, what I will see is a hydrolytic reaction. So H2O, it is hydrolyzed, splitting of the water molecule occurs, H plus and OH minus are produced. Okay. During the reaction, COA SH means coenzyme A, it is removed. This coenzyme A is removed, and this reaction, it is catalyzed by the enzyme known as citrate synthase. Okay. Now the first reaction, how acetyl-CoA means 2 carbon atoms from the acetyl-CoA, it gets entry into the TCA cycle along with oxaloacetate and it forms the citrate. Now this citrate contains how many carboxyl group? You can see are 3 carboxyl groups. That's why this cycle is also known as tricarboxylic acid cycle. I hope you must have understood this is the name why it is called as a tricarboxylic acid cycle. Okay. In the further reaction, okay, the enzyme is aconitase. Okay. Now this aconitase enzyme removes the water molecule H2 from the citrate. Okay. So from where this water molecule is removed or how it is removed? So this OH and one of the H from this CH2, it is removed in the form of water, H2O. Okay, so the structure remains like this: COOH, CH2, okay, COOH. Due to the loss of water molecule, the double bond is created between these 2 carbon atoms and COOH. This structure is called as cis-aconitase. An enzyme responsible for this reaction is called as aconitase. Okay. The next reaction, which is catalyzed by the same enzyme called as aconitase. This time it is responsible for addition of H2O. Missplitting of water molecule occurs, H plus and OH minus also ions, they are bridged across the structure, okay, across the carbon atoms. So the structure becomes what? COOH, CH2, okay, then COOH. So of the splitting of water molecule H plus and OH minus ions are produced. Now this time H plus, it is linked to this one, okay, then this on this carbon H, and here it is OH. Here it is OH. Now you can see the position of hydroxyl group. It has been changed. Earlier it was on the carbon number 3. Now it is on the carbon number 4. Okay, and remaining COOH. This is called as isocitrate. The name of the structure is isocitrate. Now this isocitrate and the citrate, what is the difference? Okay, both are isomers of each other. Structurally they are different. Structurally they are different is what? Earlier, as I said, the hydroxyl group was on the carbon number 3. Now it is on the carbon number 4. Okay, this is the difference. The position of H and OH, it is changed. Okay, so that's why they both are called as the isomers of each other. Okay, further, next reaction. Now what happens during the next reaction? The first oxidation reaction occurs in the TCA cycle or citric acid cycle. Okay, this first oxidation cycle removes electrons in the form of hydrogen atoms. Okay, and it is accepted by NAD plus. Accepted by NAD plus becomes NADH plus H plus. Okay, so 2 hydrogen atoms are removed. Okay, along with 2 glucons. Okay. Now, how and from where? During the same reaction, what happens? During the same reaction, carbon dioxide is removed. CO2, it is removed. Now, first we'll see how this carbon dioxide is removed. The carbon dioxide is removed from this carboxyl group. Okay, I have encircled this carbon COO. It is removed in the form of carbon dioxide, and a pair of electrons from two atoms are removed from this one and this one. Okay, and both are accepted by NAD, becomes NADH plus H plus. So the structure becomes COOH CH2, then CHH. So this is CH2. Now, after the loss of these two, what will remain? C double bond O and COOH. COOH. This structure is called as alpha ketoglutarate. Alpha ketoglutarate, or simply you can say alpha KGA. Okay, on alpha carbon atom, the ketonic group is created. That's why this is called as alpha ketoglutaric acid or alpha ketoglutarate. Now the subsequent reaction, in which the second dehydrogenation reaction, same thing happens. Electrons are removed from alpha ketoglutarate. They are accepted by NAD, gets reduced to NADH plus H plus, and second decarboxylation reaction occurs. Second is CO2. Okay, so I forgot to write the name of the enzyme, and that is called as isocitrate dehydrogenase. I have written the short form IDH. IDH is nothing but what? Isocitrate dehydrogenase. So isocitrate is the substrate for this enzyme, and it catalyzes a dehydrogenation type of reaction. That's why it is called as isocitrate dehydrogenase. And due to the same reaction, carbon dioxide is removed. Okay. Now let us talk about the next reaction. What is the next reaction? Or what happens to the alpha KGA? This time, name of the enzyme is alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. Now this is the enzyme similar to the enzyme which converts pyruvic acid into acetyl-CoA. The enzyme is PDH, pyruvate dehydrogenase. That is the multi-enzyme complex. Now this is also multi-enzyme complex called as alpha KDH. That is alpha ketoglutarate dehydrogenase. So during the reaction, two hydrogen atoms are removed. Similarly, they are accepted by NAD plus gets reduced to NADH plus H plus. So it gets reduced to NADH plus H plus. Simultaneously, the decarboxylation reaction occurs. Okay, as well as there is the addition of coenzyme A. Here we write addition of COASH, coenzyme A. Okay. Now, due to this, the structure becomes COOH, then CH2, third carbon, CH2. Okay. Now, fourth, C double bond O. Now this time, from this carboxyl group, this COO minus, it is removed. I have encircled that carbon. It is removed in the form of CO2. As I said, during the reaction, decarboxylation occurs, means from COOH, CO minus is removed in the form of CO2. Now, whatever the H remains, that H means hydrogen, and another hydrogen from COASH. Okay, it is accepted by NAD, gets reduced to NADH plus H plus, and this becomes SCOA. Now, what is the name of this structure? It's called as succinyl-CoA. Succinyl-CoA. Okay. Now, the succinyl-CoA further in the next reaction, what happens? This succinyl-CoA, it is converted into succinate. It is converted to succinate. down. This is unusual bond. This is called as high energy bond. This high energy bond, it is broken down. Okay, it generates a lot of energy, and that energy is used to join GDP plus PI, and it forms GDP, a high energy compound. And from the high energy compound, this GDP, another molecule of ATP is formed. Okay, so what happens during the reaction? During the reaction, COA SH is removed, and it is accompanied by hydrolysis, splitting of water, H2O, that is H plus and OH minus. Okay, so what happens? The structure becomes like this: COOH, then CH2, CO, and after splitting of water molecule, whatever the OH is formed, it becomes COOH, and H, it goes along with the COAS and that H. So that coenzyme A is removed. Now this is what the succinate, which is a 4-carbon compound, form. Okay. Now in the next reaction, what is the name of the enzyme which is responsible for conversion of succinyl CoA to succinate? The name of the enzyme is succinyl CoA synthase. What is the name? That is a succinyl CoA synthase. So succinyl CoA synthase converts the succinyl CoA to succinate, and during the reaction COASH, coenzyme A, is removed, and simultaneously whatever the bond energy is released, that is used to couple phosphate to guanosine diphosphate, and it forms GTP. From the GTP, it is converted into ATP. Okay, so succinate is formed. Now third reaction. Why I am saying third reaction? This is the third oxidation reaction. First we have seen conversion of isocitrate to alpha ketoglutarate. This is the first oxidation step in the TCA cycle. Then from alpha KGA to succinyl CoA, this is another oxidation reaction in the TCA cycle. Now third one, but this time the enzyme known as SDH. SDS stands for succinate dehydrogenase. Ultimately, the dehydrogenation reaction occurs. Hydrogens will be removed. Okay, but what happens? See, this COOH from the succinate. Okay, this succinate is converted into fumarate by the enzyme succinate granule, but this time it doesn't use coenzyme NAD. It requires a prosthetic group, and that prosthetic group is called as FAD. When a FAD accepts the electron, it gets reduced to FADH2. So how the structure becomes? It becomes like this: COOH, CH, double bond, CH, COOH. Due to the loss of one hydrogen from this carbon and another hydrogen from this carbon, so these two hydrogen atoms are removed from two adjacent carbon atoms. Okay, so ultimately there is a creation of double bond between these two carbon atoms. Okay, and then it becomes a fumarate. Okay. Now this fumarate, it is converted to malate by the enzyme fumarase, fumarase. But during this reaction, H2O is added. Means H plus and OH minus ions, they are going to add in the structure of fumarate. It will become malate. Okay, so the structure will become COOH. Second, CH and OH from the water molecule. OH H is already there, and one of the H it will come from the water molecule, so that will become CH2 and COOH. COOH. So that is called as a malate. Okay, the enzyme is fumarase, and it is responsible for addition of water molecule in the structure of fumarate. It becomes malate. And the last reaction, which is the fourth reaction, in which the fourth reaction of oxidation, in which again dehydrogenation process will occur. This time the enzyme is malate dehydrogenase. Malate dehydrogenase. It requires a coenzyme known as NAB, and after accepting the electrons, it gets reduced to NABH plus H plus. So how this, from this carbon, these two hydrogens are removed. So that's why COOH. After the loss of these two hydrogens, what will remain? C double bond O, CH2, COOH. This structure is called as oxidation. So this way, the tricarboxylic acid cycle consists 4 oxidation steps. As I said, this is the key component or key part of the central energetic pathway. Now you can calculate the number of carbons. See, acidic OA contains 2 carbon atoms, correct? This one, 2. Okay. So due to the addition of these 2 carbon atoms along with this 4 carbon structure, oxidation, it is responsible for production of this 6 carbon citrate. Okay. Further, it is converted to 6 carbon cis-aconitate. Further, it is responsible for production of 6 carbon isocitrate. From isocitrate, one of the carbon is removed in the form of carbon dioxide. Okay, so alpha Kg is formed, which contains 5 carbon. Okay, from alpha Kga, again the decarboxylation reaction occurs. One carbon is again removed in the form of carbon dioxide. Okay, and the structure formed, that is called the succinate OA. Now this contains 1, 2, 3, 4 carbon atoms. Okay, and from this the succinate contains 4 carbon, fumarate contains 4 carbon, malate contains 4 carbon, and oxaloacetate again after recycling it contains 4 carbon atoms. So whatever the 2 carbons they were introduced from the acetyl OA into the TCA cycle, those are removed in the form of carbon dioxide in these 2 steps. Okay, and simultaneously the oxidation occurs at 4 steps, and those oxidation steps releases the electrons. Okay, and those electrons are accepted either by NAD or FAD, and finally it donates the electrons to electron transport chain and the energy is generated. Okay, this is nothing but what a TCA cycle. I hope you must have understood with the structures, whatever the change occurs during the reaction, which enzyme is involved in this, what type of change occurs, and what is the overall cycle. That I hope you must have understood. Okay, thank you very much.